Hey, good morning, guys. I know this is the last panel before lunch, so there's a lot of great additional information. Really great program. Evan, congratulations so far this week. It's really been remarkable. My name is Steve Wolf. I'm the president and co-founder of the Beyond Earth Institute. It is a nonprofit, nonpartisan think tank based in Washington where we are focused on human spaceflight and human habitation. So that's very apropos to what our panel is all about today. We are looking at creating the legal and policy framework that will enable this distant future where humans will one day become multi-planet species. You guys have heard those visionary thoughts by giants of industry like Musk and Bezos. Well, how do we make that future a reality from a policy standpoint? That's where we come from, and that's my perspective here. In this program, which is titled Humans in Space, Health Habitats and Commercial Pathways, we are very fortunate that we have a panel here of folks that are working at the right next to the tech, particularly in the bio area. They are working, they're not policy people, they're not high-level people, so they're the guys that are in the trenches really getting the research answers that we need in order to answer the big questions. So let's talk, so to set this up, NASA has 65 years of experience with humans in space, especially with the ISS program. However, there is still so much that we don't know how to keep humans alive for long duration. So NASA has in their human research program, they have 175 gaps in technologies relating to human abilities to survive in space, microgravity, radiation, and so forth. That also equates to 1,342 different tasks that they have to complete in order to answer all those questions. So the path to understanding this is quite substantial, and that's where our guests today, our panelists today, are gonna get involved. When you talk about bringing those cancer cells back down to Earth, you're talking about an application that's going to maybe drive cancer research for the benefit of everyone, you know. So, but Jangkeun, what's your thoughts on this topic?
Yeah, with the commercialization, from several years, the most of the bottleneck coming from the accessibility, but with the commercialization, mostly I agree with Devon and Jeremy. The bottleneck was the accessibility, and with the commercialization, it opens up that bottleneck, but it does not mean only for the scaling itself. It opens up kind of new way of science. So with the in-flight experiment, there is no gravity, so the pharmaceutical company or researchers try to utilize the without gravity environment for some protein crystallization to have a better structure, which will lead to better drug discovery or accelerated drug discovery, or for regenerative medicine without gravity. The organoid or some stem cells having another opportunity to basically is getting bigger, easily to make it bigger organoids, so it'd be helpful for the regenerative medicine and also aging and muscle degeneration of bone reduction could be accelerated. So if we could find some mechanism or clue from space, we can adapt it to the Earth for the kind of longevity or health span.
Mm-hmm. I'm going to ask you a question in a second, Jeremy. By the way, I don't want to wait till the end for question and answer questions, so let's just do it as we go. I just saw a hand shot right up. She's dying to ask a question. Go
I'm
ahead.
so sorry. I don't mean to hijack. So an earlier panel mentioned international alliances and partnerships and the British 3-star with our 4-star. Has anybody explored Demis Hospice, who was using AI and he just solved all the DNA sequences? There's a documentary, The Thinking Game. He also was bought out by DeepMind, AlphaGo, Google. So have you worked with anything like some kind of partnership, international alliance with that? Just throwing that out there if anybody's done that research.
Are you familiar with?
Demis Hospice, he's just knighted and kinged and crowned and all of his jazz. He was the one who wouldn't come to Silicon Valley. He stayed in London. But he's got a deep and wide history and bench with AI and forward thinking, and he beat AlphaGo, Go.
Well, let's. Are you guys familiar with this? And if not,
I believe most of people know the DeepMind and the AlphaGo and Demis Hospice, but currently I'm not closely worked with kind of adapting some AI technology into the space biomedicine, but there are some multiple groups and multiple pitches trying to use the AI as an agent. As an agent for kind of in labor space, we need there is no kind of doctor at there, so kind of developing some medical AI agent or some helping with predicting who can go to the space or who can be more fit for the space. But so there are some kind of AI companies trying to utilize our database coming from ChemBank, from Jeremy or Astronaut Database to train some AI model, but currently we are not closely working with them yet.
Okay.
but let's stay on that question, if I might just broaden the lens there, or just speak to, the AI as a tool for the analysis you're doing. You're collecting all of this data. One of your experiments, you're collecting 74, you're collecting quite a bit of data, and how are you utilizing AI? Is that helping you?
Yeah, so I'll speak to the complexity of the data sets that we're talking about. I spoke in my introduction about, part of what we're doing is multi-omics, right? So genomics, proteomics, metabolomics, trying to take all of these different slices of biology that historically have been kind of viewed as their own kind of area of study and integrating them to try to derive larger systems levels conclusions. That's not straightforward and requires a lot of high-powered computing. And so I'm a bench scientist. I'm not really on the computational side. I'm really good at making a lot of data and not really great at dealing with it once it's created. I have people like Jangkeun helps a bit more with that kind of stuff. But AI is going to be increasingly valuable in terms of integrating data sets from different sources and deriving those larger conclusions for sure.
Okay, last thought I'll just throw in.
I think this isn't working. Oh, he has an open source model.
He gave it to the world, so it's something that could be farmed or a data lake or whatever it is.
Yeah,
Just throwing that out there.
Being an academic institution, we are extremely collaborative and do work with a lot of research groups across the world. One of our biggest packages of papers included over 100 institutions from all around the world. So we're very quick to work with other people and love the idea of data sharing and open source platforms and stuff like that. So definitely look into that.
Devon, you have a thought to that?
I'm also a bench scientist.
But I don't think there's any doubt that AI is going to drive your research just like it's going to drive all data research that's happening right now. So Jeremy, I want to come back to you. Inspiration 4, you were involved in that mission that involved our current NASA administrator, Jared Isaacman, which is super cool. And tell us about that program and the milestones that you achieved and where you're going from here on that.
Yeah, sure. So we have since replicated what we did for that to some capacity for a couple other missions, and I think that it was the first of its kind in being a academic study around this commercial crew.
Explain the experiments.
Yeah. I mean, even just the act of collecting samples, which for me that was a big role I played. That was ultimately what kicked off the beginning of what our biobank ended up being. Is we had the opportunity to get these samples from these astronauts, and that required us basically to travel around the country to where the astronauts were going to be and collect the samples from them at that time. And that's not a very scalable model. And I think what we've learned from that is we can get this done. We can, hack up a lab into a small transportable thing and go to Florida and Houston and California and get these samples. But at the end of the day, it was the biggest collection of space data to be published in one package, and yet there was an N of 4 for that mission. And so there were some really cool findings that I think were definitely interesting as a case study, but it is really just the kicking off point that if we want to achieve statistical power and say any of these findings are generalizable and applicable as we bring them down to earth, we're going to need to scale these operations. And the way that we're achieving what we're doing now isn't the most scalable. And so that comes into how do we advocate within the industry to build out the infrastructure that allows for sample collection, sample processing, and sample storage to be standardized and efficient when it's happening in so many different locations.
So, and I don't know if we're going to back up here a little bit, but the biobanking. So, give us a little more context on what that is and what your hopes are for that.
Yeah, so biobanking has kind of become a gold standard of medical research now, because you basically can take an individual sample, be that a blood tube or a tissue or urine, and fractionate that single sample into many allopons that are all treated identically. And so then you have standardization across these samples and a backlog of samples that if someone's doing research and comes across your study and says, hey, that's really interesting, I wish they did this study, now there's samples available that they can request that aren't just similar, they are the same sample. And that really is how we develop statistical power around our findings. And so basically, there aren't that many people that go to space. So if you have an opportunity to collect samples from some, you should be getting as much as you can and storing it like it's gold, because that will pay dividends in the long term. As I said, the I-4 mission had an N of 4, but as we continue to do these collections and build out our biobank, we now have 14 astronauts represented within the biobank that, yes, there are confounds in terms of comparing across missions, but we're doing a lot in terms of building up what we can take away from a really small set of people. And so collections for I-4 and the subsequent missions, we have an opportunity to get samples. We will fly out to them, partner with a local lab, and collect as many samples as they are willing to give us. So that's blood, stool, urine, and then take it to the lab, process it, and send it back here to New York on dry ice to get into our bank.
One of the, I think we're going to come back to this, but one of the things we hear in Washington is, we can't get enough data, right, from human presence in space and the kind of data you're talking about. But there's an issue of individuals going to space being willing to subject themselves to that kind of data collection, because sometimes it's a little intrusive. They have to they may have to do maybe some blood draws or maybe some wear electrodes and so forth. And you mentioned stools, which could be in space. How do you collect the stool? But and some people are suggesting that there should be legislation that says that if you're going to be a private astronaut that's going into space, you're going to agree to subject yourself to a degree of research so that the information your body provides will help us in the future to improve the safety of being in space. You're shaking your head and you're saying that that's a good idea?
I'm personally a fairly big advocate of that. I think that historically the field has been somewhat militaristic in how they view astronauts in terms of there's a mission and we want to get it done. And if something happens to you during that mission, we'll do our best to characterize it, but the primary goal is to get the mission done. And I think, especially as we start sending more and more people to space, our understanding the health of the astronauts and what's happening to them when they go to space, considering that they're putting themselves at remarkable risk in such an extreme environment, should be at the forefront of prioritization in terms of mission goals. And so legislation like that could help push us forward.
Jangkeun, do you have an opinion on that? You want to offer
Yeah,
a thought?
mostly I second Jeremy, so, yeah. But, yeah, like this, we are collecting samples from clinical trials, so for some, I think we can learn from how we collect the samples and how we utilize that one in a kind of clinical setting, so mostly we can, need some kind of agreement across the society, but, you know, we can keep, our group can keep asking, keep explaining how we can learn from this kind of thing.
Okay, thank you. And Devon's going to jump in, and then I'm going to have another, we're going to switch gears after.
Sure,
your
yeah,
statement.
I'll just be a little bit of a dissenting voice on that. So I personally, I think bodily autonomy is very important, informed consent, obviously very critical. So I'm not sure personally that I would support a legislation like that, just in terms of, I think, all the people that are going to space should have the decision about how their data is used, and something that Jeremy alluded to earlier, where there are barriers where NASA as a government agency will, despite perhaps astronauts giving full informed consent, NASA as an agency will step in and say, no, we would not like for you to use these samples in this way. So I do think there's room for maximum informed consent for the participants, but also less government intervention in terms of when somebody does consent, how that data ends up getting generated and used. So I do think there's a little bit of nuance there.
And we also know that astronauts have the right to refuse certain kinds of invasive.
Yeah, if I might just add to that, I think the pressure should be put on the launching organizations. The companies should be required to make monitoring astronaut health a primary of their mission. If astronauts then don't want to consent to certain aspects, I totally agree that they shouldn't be required to do that.
Yeah.
But the companies need to start focusing on this, especially if we want to put people on the moon and Mars for long term.
You guys are helping me, so we're working on the legislation now. Question here.
Hi. So I have a question. So you guys are using commercial astronaut biospecimens. And so I'm wondering, I guess, comparing it to like a government astronaut, trained astronaut, what do those are you collecting before launch, during launch, and then after launch? And I guess if you looking at a commercial astronaut, I'm interested in seeing what that looks like versus someone that is a government astronaut. Are those the cellular pathways are different, or the protein? Because how long are they up there? They're not trained like a NASA astronaut who might be doing a year-long space mission, where I would assume that the radiation up there and the gravity up there is impacting them way more than a commercial astronaut that's up there for a week or something. I don't know.
Maybe I can explain, and maybe Jeremy or Devon can back up. So our team has an opportunity to get some data from the NASA Twin study, which was a year-long mission, and some of the sample from the short-term and mid-term mission. Short-term would be less than 3 months in space, and mid-term would be around 6 months. So the major difference between the government mission and the commercial mission is the duration of the mission. So normally the NASA astronauts stay in more than 1 month, and average is between 3 to 6 months. And commercial space flight is with 3 to 10 days. So Inspiration 4 was 3 days. Polaris Dawn follow-up was 5 days, and Axiom 2 mission was commercial space flight ascent to the International Space Station, and it's 10 days. So the most striking thing was that when we compared to 1 year mission of Twin study with the short-term data, the most of the changes are consistent, even though it was 3 days and 1 year. So telomere is a kind of end of the chromosome. It is getting longer in space. In Twin study was striking, but was reproduced in the 3 day space flight mission, and immune cell changes or some brain-related changes, mostly and mitochondrial change. Mitochondria is kind of a cell organelle making energy. There is a kind of consistent changes from the spaceflight 1-year mission at 3D. And when the mission is big, there are some model organism experiments. So scientists send some mouse or Drosophila model organism sent to the space. And we have a shared data set in the Gene lab from the NASA. And also when you compare, there's kind of consistent changes around the mission, species, cell types after the space flight. So that's kind of interesting thing.
Just out of curiosity, sorry, when they return back, do you see those changes, like telomere changes and stuff, or even, like, say, like a month after, or something, or is it still stay the same?
That's a great question. So that's why Jeremy is keep tracking long-term sample biobanking. So most of changes happen rapidly when people go to the space, and it recovered those rapidly when they return to Earth. So kind of return day 1, 0, 1, there is still some changes, but kind of after 7 days, 2 weeks, when we followed up, most of changes recovered very rapidly. But still, there are some long-term changes over 3-6 months, but it's kind of a little bit marginal.
Oh, awesome, thanks, guys.
Well, it does seem like there's a lot of, what we need to understand is what does this really mean? And what is that in terms of potential? I want to talk about telomeres and the length of them over time and in terms of aging and all this. But so that'll be interesting to see where that research goes. But I want to switch now, if I could, I'll come back to you. I want to switch, I want to talk about radiation, and this is where Devon is doing a lot of research in this area. He's at the intersection of space radiation and cancer biology, which is super cool. Tell us about what you're doing and how this can help people on Earth.
Yeah, so Memorial Sloan Kettering cares exclusively about trying to cure cancer on Earth and hopefully in space, if I can convince them. And my background was doing breast cancer research at CUNY, at Hunter College here in the city. And I transitioned about 5 years ago to be at Sloan Kettering. And one of the major things I wanted to do, I grew up in Orlando watching the shuttle program and seeing the twin study throughout my PhD, and I realized we're in an era now where you can do space biology and interact with humans and cells, and there's more access than ever. So actually Jangkeun and I met at a Humans in Space program hosted by Axiom Space and Orion, and we both pitched and won private equity funding to do a little bit of our research. And my project was geared towards looking at how space radiation is damaging DNA and how we can protect astronauts with a bunch of different pharmaceutical interventions, which can be used hopefully as a standalone, but ideally in combination with things like increased radiation shielding and other physical barriers. So we actually went to the Columbia Neutron Beam Facility. They have, I forget the acronym, but it's called RARAF. It's like the Radiological Accelerator Research Facility, something like that. And they're a great facility up in Tarrytown. They have a big particle accelerator that sits in the basement. Looks like this big Soviet-era machine. And you can load up your cells into it, shoot it with space radiation, one of the very few places in the US and in the world that can do space radiation on Earth. And we tested a bunch of pharmacological drug interventions to see whether we could reduce DNA damage, and we have been able to. We haven't quite published yet, but one of the things to recap on the telomeres that we found really interesting was that we're not in a spaceflight environment. We're on the ground. We're in Tarrytown. We're shooting cells with space radiation, and we see lengthening of telomeres as well. So trying to parse out all the different multivariables that happen in space, whether it's microgravity or radiation or isolation or whatever, you can start to try to tease these things out. I mean, scared to move. Okay, good. All right, good. So one thing that is unpublished, but that we're kind of trying to work out and tease out with some of these other PIs like Chris Mason and Susan Bailey is the lengthening of telomeres a function exclusively of radiation? And I think Susan Bailey has also shown high altitude climbers, when you go up to Everest and back, also have kind of longer telomeres. And you're getting increased cosmic radiation with altitude, whether you're on a commercial flight or climbing Everest or on ISS or in Tarrytown, I guess, in front of the particle accelerator. So that's the fast version, and all of this is immediately relevant to radiation therapeutics on Earth, trying to improve where we deliver radiation for cancer patients and the quality of that radiation as well. How can we use that to better understand DNA repair and better treat patients here on Earth?
Well, I have a follow-up question is on, when we talk about radiation protection, we usually talk about shielding, right? So you're talking about using pharmaceuticals to protect us from this. What do you anticipate in the future that it'd be some combination of that for the astronauts to protect them long-term? Other speculation is that just humans being in space over time may actually develop a radiation resistance. I mean, do you give any validity to those kinds of hopes, I guess?
Yeah, sure. Yeah, interesting. So if you've taken any radiation training, you'll know this. If you haven't, I'm happy to teach you today. The three big things to limit your risk in radiation exposure is time, distance, and shielding are the things that we talk about. So when you're talking about spaceflight missions, if you're in space, you can try to limit the amount of time that you're there, right? Your distance is not really modular. I mean, the closer you are to Earth's magnetosphere, the more protected you are, but if you're going on a moon mission, whether through the Van Allen belts or just further from the magnetosphere, you're getting more radiation dose. And shielding is perhaps the biggest component of that, but the higher energy spectrum of cosmic radiation as you leave the Van Allen belts is such that you need about a swimming pool full of water or 12 feet of lead to shield, neither of which particularly cheap to launch in current state. Even if Starship launch costs come down, people don't really want to launch a swimming pool full of water. There are big advancements in material science and composites and trying to, another great New York company, Melogen Labs, who I have no financial relationship with, but are developing shielding technology to help improve particularly satellites, but also potentially humans and biology. So I think it'll be a combination of kind of pharmacological intervention and better composites and materials. I don't think that humans will get more resistant to radiation unless you're talking about a time scale of like hundreds of thousands of years. So I don't know that that's realistic, just from the perspective of just from the perspective that DNA is quite fragile, and there are organisms on Earth that have evolved to be radio resistant, like radiodurans. They found near Chernobyl can kind of withstand and possibly metabolize radiation, but we are not close to that. Some people, like the Mason Lab, would like to do, maybe I'll let you talk about this, but would like to do a synthetic biology approach and sort of, I'm also not a big proponent of this, but genetically engineer humans to be more spacefaring.
Where it was going.
Yeah, so I'll leave it, I'll pass it to Jeremy with that. Let's talk about it.
Let's not dig deeper into that,
Okay.
unless you
We can
really
talk about it
feel like
once.
you need to, but we are starting to run out of time, and I realize I still have a bunch of questions here, but I do want to bring in the aspect, well, two aspects. One is where is the intersection with what you guys are doing and developing commercial or business models around what you're doing so that this could be an ongoing private investment opportunity for yourselves or others. And then I guess I'll just throw it in there is the New York angle. I mean, you guys are, New York seems to be becoming is a hub for bio research, and so it seems like we're spinning off of that opportunity. But speak to those 2 points if you can. Go ahead.
Maybe I can do quickly. So first, kind of business model kind of thing. So what our teams in Chris Mason and Jeremy and our collaborator in Pittsburgh, it's not it's not kind of exotic one. It's actually, we learned from the astronaut or space flight or space analog. So there is a simulated radiation or microgravity, and we found something, and with those discovery, we start to another kind of clinical trial. So we are collaborating with Otsuka Pharmaceutical, is one of the leading pharmaceutical company in Japan, and the CEO is found our research and very eager to support that idea. So they provide their one of the nutraceutical, which could be used for protecting some changes from the spaceflight we found. So we started some clinical trial or preclinical ideas, and in the long term we want to bring those the drug into the space. And actually at FREM2 last year there was a kind of opportunity we could bring some in vitro experiment. It's not human experiment, but it's a cell or some other organism experiment, and we brought that chemical to test if the drug can protect some changes from the spaceflight. So it's not far from the pharmaceutical or oncology business. So we learned from space, and we could utilize that experiment for the art. So that would be one of the model. And for the second question, as our team started the biggest biobank for the astronaut and the database, and we know there are a lot of great researchers and hospital and institute and some startup ecosystem in New York City. So given our the database or sample accessibility, if we could make some kind of interconnected ecosystem with hospital, institute, and the startup, there would be something interesting thing we could do as there are more accessibility through the commercial space flights, space station.
Great, yeah, no. And we look forward to hearing a lot more about the biobank, which is a very exciting project. Did you want to add to that, Jeremy?
Sure, let's see this. Oh, yeah, let's do that. Yeah, can you hear me okay? Yeah, I mean, so personally, as far as the commercialization aspect goes, I think a lot of the work that we're doing right now is foundational and will set us up to take advantage of kind of the next phase of development for research platforms in orbit. So there, as Jangkeun mentioned, there's a couple of commercial space stations that are planning to be launched within the next several years that ultimately will provide basically an analogous kind of agility that commercial space has allowed for our research to an on-orbit platform like the ISS. And I think what we can learn from what we're doing now will enable us to take advantage of those kinds of platforms. I think as far as product commercialization, most of what we're going to see initially is going to be biomaterials, really, as opposed to biomedicine. So, for example, there's companies like Lambda Vision that's planning on, they're partnered with Star Labs, which doesn't exist yet, but when it's in orbit, they're going to basically print synthetic retinas that theoretically are like far better because they're printed in microgravity. And that is a type of biology, but it's more of a biomaterial that then gets brought down and applied. And that's where I really see a lot of commercialization opportunities.
Space manufacturing.
Exactly.
Yeah.
And but just to touch on the New York side, I really think the competency of New York within medical research, first and foremost, but also just as a centralization hub. For me, a lot of what the frustration and like the holes of our logistical capabilities are is just how decentralized everything is. You know, we have these massive governmental organizations like NASA, when they're controlling everything for their medical studies, they get to centralize everything. Now with these commercial efforts, there's no centralization. We're an academic group. There's several other across the country that are trying to do similar things, and if we could work in a centralized fashion, we'd be far more efficient in terms of what we could achieve, and the fidelity of the samples would be far better. We're not shipping things across the country or repeating research. There's a group that's doing very similar stuff to what we're doing. All of those things, and New York is great at centralizing things, and making it like a hub could be a really effective way to advance the field.
Thanks, Jeremy. We are already out of time. I do want to give Devon the last word so that we can get you guys to lunch. And sorry for the folks who had questions.
Yeah, we're happy to chat with people at lunch too. I'll make it quick, and I'll try to make it quick. So yeah, I think that in-space manufacturing and all of that stuff is a great story, including kind of Merck and Keytruda. There's a great article that just kind of came out about, is this the next era of space manufacturing? And maybe the answer is not necessarily, but it can teach us things about how to manufacture better on Earth. So people like Varda and other really large companies that are in the space pharmaceutical world are looking for this kind of magical, mystical compound or material that can only be manufactured in orbit. So far, it has not been found, and even if we found it today, the regulatory pathway is such that it would be another 10, 15 years before it gets to market. But I think where New York is kind of uniquely situated is we have the talent, we have the capital here, we have a lot of eyes and attention on what's going on, and so we may not have a launch facility, but we do have a lot of really awesome stuff going on here. So I'm happy to talk about any of that at lunch and afterwards, and great to be on the panel with you. And if you've liked anything you've heard today, I will be on the job market soon, so I'm happy to entertain any conversations about that.
Well, with that, let's give a round of applause to our panel. Thank you all for your interest. And I know